Thank you, Nadir. Thank you for this invitation to be here and to meet in such a big crowd. So today I would like to give you a quick overview of the challenges and the issues related to the management of DNA data related to museum specimens. I would like also to give some solutions and existing solutions, how we think to implement them in the, sorry, there's an echo, no? Okay, it's better. You hear me? Okay, so how we think about solutions and how we want to implement them in our current SwissCollNet project on biobanks in Geneva. So I know that genetic data is not maybe the main focus of SwissCollNet, but still I think it's important to consider this category of information within this context. Now, more and more genetic data are used to study biodiversity, and it's not stopping. It's going to increase. This dynamic is going to increase probably because of the new techniques, the genomic techniques, and new fields of research like museomics. So in this context, the specimens are really important, key elements. Specimens in museums can provide a direct source of DNA. They are also frequently used to as voucher specimens for DNA data to check the source, the origins of the information we analyze. So DNA data deriving from specimens are now really largely used to reconstruct the tree of life, with direct impact on taxonomy and systematics, on names we put on diversity. This kind of information is also used in the context of barcoding of life to build reference. Specimens are used to reference these barcodes. And more specimens are also used to monitor the genetic diversity. There are now ongoing projects like the project led by Nadir Decline or at ETH Geneva project. And now we are also able more and more easily to sequence at large scale, like full genomes, like in the consortiums of ERGA, they're active into this field, and also in the context of larger European projects like Biodiversity Genomics Europe that just started. So all this information is going to also to implement, to change a bit our view of biodiversity, and as a direct also impact on names, on things that we put, how we curate the collections. And so it's important that there's a smooth communication between these two boxes. So for the moment, the information related to specimens is stored in GBIF, that we know that. Information related to genetic data is more into other databases like for barcodes, BOLD, or in more general context, EMBL, NCBI handle these, store the raw data associated to DNA. And more processed data like assembled sequences or alignments are also sometimes stored in more disparate public repository like DRIAD. So database exists, the structure exists, but what's the problem is more the communication between these two type of databases, which is, there are some solutions, but this for the moment not very satisfactory, the links between these two types of informations. And this creates some problems, some limitations. I want to try to illustrate these limitations by addressing some easy questions you might be interested to solve in the context of your institutions. Like if you want to know, for instance, how many type specimens hosted in the Swiss collections have been sequenced. It seems obvious, so it seems something you could be interested to a number, but it's quite difficult to have a global view of what has been sequenced as type specimens, for instance, or any other kind of specimens, type of specimen. If you are interested by specific regions or locality, if you want to know, for instance, what are the genetic data available for the alpine fauna, to identify the gaps or to perform a meta-analysis of the existing data, is something also, for the moment, with the quite difficult to have a complete view of the resources. And another example, if you are interested to use a sample or DNA extract that is already available for one species, like the Edelweiss or any kind of other species, and it's also difficult to know, to have a global view, because there is no DNA bank, national DNA bank available in Switzerland. So, yeah, why is it the case? So, yeah, the conclusion is that there is no, for the moment, currently, no easy automatic way to answer these questions. And just to show you why, one example why it is not the case. An example from my research. It's a recent publication by Angela Cano, a former PhD student in my lab. And so she was interested in the evolution and origin of palms in Central America. She published a paper last year, and she extensively collected palms in Panama, this highly diverse country. So these palms are, these collections are stored in our collections. The database are available in GBIF. That's fine. She also generated a lot of next generation sequences. So these data is available in GenBank. So all the data is safely stored and organized. But if you are in GenBank and you want to go back to the specimens, it's very difficult to reconstruct the path. You have to go to the annex and to download the table, and then to be able to reconnect to the specimen. And if you are in the collection or in GBIF, it's also difficult to go to access to where is the DNA data, and to have a link toward this that point to these resources. So there's a kind of broken link in both directions that make the connection between specimens and data, the source data, the data that is generated from these specimens difficult. So it's and I would say that we respect the rules and the guidelines how to publish that. So it's not the question, but we can do a better job creating these links. And there are solutions that exist to do that. The GBIF just published guidelines to how to deal with DNA-derived data. There are also dedicated database like GGBN, Global Genome Biodiversity Network, that deals with DNA and tissues. And now it exists also a set of Darwin core extension, the Darwin terms, to deal to structure data, the data deriving from DNA. So I really encourage everybody to use now these terms so that we speak a common language. And in GBIF, we can also now also have an access to the data, DNA data. It's possible to create links that redirect you from GBIF to where the data is stored. Is, for instance, a case for all the barcodes resources that is now available also through GBIF. But for the rest of the markers or DNA data is less this links is less frequent. There also the development of new tools to database this type of information to make these links, like Geome, for instance. It's interesting to consider, to be inspired by these initiatives. And just to mention that at the national level also there is now a current project, an idea to develop a kind of national database that gives you access to this, to the genetic data at the national level, like GenDib in VSL. So there are some solutions, already some resources available, and well, we need to use them and to make. And for that, we really need to speak about, to adopt the same standards and vocabulary, the Darwin core. I think you understand that today, and so that we can really implement this. And I will just to show you how we think about it in our lab, how we try to create a DNA bank. And I do that with Guillaume Antonioli. He's here also in this room today. So he's participated to the brain thinking, to the creation of these tables. And our starting point is a specimen stored for instance, in our herbarium with a unique identifier. So that's the starting point. And now we want to link these vouchers, these specimens, with a new categories of data. In the context of our DNA bank, so we can have tissues associated to specimens or DNA extracts, and we listed the tables of these two tables that describe the identity of these items and where they are stored, and for DNA extracts also the quality and quantity of the DNA we can have. So that's things that we anyway record, information we record in our lab, but now we found some terms that is in blue that are Darwin core. So there are standards with vocabulary, defined vocabularies, and I think it's really important so that we can now share this information with the Swiss node of the GBIF and with databases like GGBN. And well, usually we don't stop with extractions, but DNA extractions, but from that we generate data sequences, and we have also some tables that now to list the terms that will help us to record information associated with either PCR or genomic libraries, and then to the end products, which is the sequences. And the thing is that everything now is connected, and from the endpoints to the end products, like the sequences, you can trace back the informations and go back to the source specimen. So in blue, again, it's Darwin core terms that could be exchanged in the future. In green is the terms that are more deriving from EMBL, gene bank, and that could also serve, these tables could also serve to directly export and publish the DNA, these informations to these databases. At the end, we can also list where the sequences are stored and which repository, and especially the associated sequences term is something that could be provided in GBIF to point to where the sequence is stored. So it's, for the moment, it's where we are in this project that just started. So we define these tables, and if you have some suggestions, some ideas also, and if you are interested also to use these structures, we are happy to share it and to know what you think about it. For the next steps, so we will Now, the first things to do is really to complete the databasing of specimens used for genetic studies at G. So usually in this process, what's missing, unfortunately, is the recording of the source specimen used for the sequences. So we want to complete that in the first step. To structure of DNA data, so we have a collection of many spreadsheets for the moment from master students, from PhD students that created their own data spreadsheet with their own structures. And so we want to uniform this and adopt these structures to aggregate all these disparate spreadsheets and to fill the tables. So then we have to think about the mapping and how we want to transfer this information to the Swiss node of SPIF and to the GGBN. There's also the idea to develop a Botalista module to facilitate the data captures and the management of this data. And we would like to share these informations and tables for our users, so we can maybe create a community of institutions, of users that are interested in creating a DNA bank in their institutions. And I think it's important also maybe to enrich or to integrate this DNA-derived data within the Swiss data aggregator. And it would be good also maybe to think about to give access to this type of data through the Swiss Virtual National History collections in the future. So with that, I would like to thank Guillaume Antonioli that really was active in this project, and my colleagues from the Botanical Garden of Geneva, and Sophia Villers and Pascal Choudin from the Swiss node of GBIF. Thank you.
Mathieu. We have time for 1 or 2 questions for Mathieu. Who'd like to start? Yoyan.
Hi, thank you very much. I think this is very important because it also shows the relevance of natural history collections for genetic work, for instance, and makes that trackable. But that applies to many fields, also, for instance, plant morphology. And there's this big database of plants, a tri-database that links information via species names. It's terrible. So in the broader context, my question is, do you think that adding modules of Darwin Core forcing users to use Darwin Core is that expandable, like making bigger for other types of data derived from specimens as well, ecological, morphological, isotopes, etc.?
Well, for other categories, we kind of discovered these Darwin Core terms for DNA, so we were quite ignorant about this, so we had to see what was available. And I must say that for images or for other fields, I'm not. It's by extension, so I don't know exactly for other fields, but we were lucky that now the DNA-derived extensions is available, so that provides a set of terms that So it's not really revolutionary, but just because these terms are matched with the categories of fields that we already consider, but it's just the fact that we use really the correct terms. So I guess, for the other, I think that I'm not an expert on GB. Maybe Pascal or could answer these questions. Yeah, so.
Is there any other question, Michel?
Thank you. No, it's just to say that this particular issue, is actually being worked on at 2 different levels. So there's a new research infrastructure that's just been created in Europe that's called DISCO, the Distributed System of Scientific Collections. And they're really using the concept of extended specimens And in the concept of extended specimens, everything is linked to a physical object. So to the physical object, you link DNA, photographs, morphology, traits, and field information. And this is really essential. So I think it's good if you can check into the DISCO network, and that this stuff is kind of being worked on currently. And also in parallel with that, GBIF is also looking at this extended specimen model, so that, again, everything is linked to the individual specimen. So rather than turning it around and doing DNA first, it's really DNA becomes part of an extended data set linked to the physical object. And that could be, you know, like the DNA plus an analysis or data or field notes. And this is really interesting because it means that for any one specimen, through that unique identifier, you can get to a whole bunch of other information. And that's kind of going back to Jurien's point, and the issue here is it's really an exciting time because we have to think about these things now so that in the future we have all this linking done. But we can talk about the DISCO later, but this notion of extended specimen is becoming really, really important, and having everything linked to everything. So you can call up a specimen and get to other things that are connected with it. And this is all going through the codes, the standardization of the data, and all going through the unique identifiers. So having those codes on stuff is really important because the machines can read those and make relationships. So just, it's active right now. It's good news.
Yeah.
Thank you.
So I think that there are exciting developments in going on. But the first step is really that we for that to happen, it's necessary to use the terms and to adopt a common language. So, yeah.
All right. We have to move forward, but thank you very much, Mathieu.